Good morning. Now we look at the third aspect of cancer, that is apoptosis. Now, apoptosis is a pathway of cell death. It can be also regarded as a tightly regulated suicide program. Now, cells which are destined to die, they activate certain enzymes which are capable of degrading its own cellular DNA and cytoplasmic proteins. Interestingly, the plasma membranes of apoptotic cells remain intact, but the membranes are altered in such a way that the cell and its fragments become targets of phagocytosis. These dead cells are then rapidly cleared before its contents are leaked out. So the cell death occurs very quietly and does not produce any inflammatory response in the host. So as you can see in the figure, this is a very simplistic aspect of apoptosis. The cells which are targeted for cell deaths, what happens? You have chromatin condensation. There is no alteration of the membrane in the sense that the plasma membrane does not break. It still remains intact, but it undergoes what is known as membrane blebbing, and ultimately there is degradation of the cells and apoptotic body formation, and these are then cleared out before any contents are leaked, and so there is no inflammatory response in the host cell. So apoptosis on one hand is a normal physiological phenomenon. Normally what happens, it is required to eliminate cells that are no longer needed and to maintain a steady number of cell populations, particularly during embryogenesis and organogenesis, etc. In pathologic conditions, however, it may be required to eliminate cells that are genetically altered. That is, the DNA may be injured beyond repair, that is, it may have undergone the effects of radiation or cytotoxic drugs, chemical carcinogens, or extremes of temperature. What is the mechanism of apoptosis? Apoptosis is an active enzymatic process. The fundamental event is the activation of the enzyme caspase. There are two distinct pathways for caspase activation. One is the mitochondrial pathway, and the second is known as the death receptor pathway. Now, the most important pathway is the mitochondrial pathway. The mitochondria contains several proteins which induces apoptosis. These proteins are able to remove cytochrome C. That is, cytochrome C receives a signal from these proteins and it escapes from the intermembranous space of the mitochondria to the cytosol. Here it interacts with what is known as the apoptotic protease activating factor 1 and forms an apoptosome which participates in the activation of the enzyme caspase. Now what happens in the death receptor pathway? Many cells express surface molecules called death receptors. These death receptors are also able to trigger apoptosis. One example is a member of the tumor necrosis factor TNF receptor, and this pathway is also involved in the elimination of self-reactive lymphocytes. So this is the mitochondrial pathway. We find that there are several proteins which receive the signal or the apoptotic signals. They release certain apoptotic molecules or proteins which are called as BAD or BAX, BAD, BAX, etc. They alter the permeability of the mitochondrial membrane. The pores are opened up so that the cytochrome C moves from the intermembrane space to the outer membrane and exits into the cytosol. Once it exits into the cytosol, the cytochrome C then binds with the certain proteins there, and it forms what is known as the apoptosome formation occurs. And this apoptosome then activates the enzyme caspase, and the cascade will start. This is also known as the intrinsic pathway. Now this is a more detailed explanation of the intrinsic pathway of the mitochondria. Now, receiving a signal for apoptosis, certain proteins are released by the mitochondrial membrane, which is known as the free apoptotic proteins. These apoptotic proteins are also under the control of certain are also part of the BCL2 family. The free apoptotic proteins are also part of the BCL2 family. Some of the members of the BCL2 family are pro-apoptotic. So it releases pro-apoptotic proteins. These pro-apoptotic proteins are able to change the pore conformation of the mitochondrial membrane so that the cytochrome C is released from the intermembrane space to the external membrane, where it reacts with certain proteins known as the APAF1 proteins to form an apoptosome. The apoptosome, once it is formed, it is able to activate caspase 3. So the APAF1 is the apoptosome activating factor 1. So the cytochrome C binds to the apoptosome activating factor 1. It forms an apoptosome. The apoptosome then activates the enzyme caspase 3, which then activates the caspase cascade, resulting in cell blebbing and the final collapse of the cell. Now the second pathway is known as the extrinsic pathway. As I told you, in the extrinsic pathway, the certain cells have certain receptors on the membrane which are known as the death receptors. When the signal for apoptosis occurs, there are certain pro-apoptotic ligands which bind to these death receptors. So this bound pro-apoptotic ligands with the death receptor then activates the enzyme caspase. The activated caspases then activate certain nucleases and proteases, which then cause the death of the cell, that is the apoptosis of the cell. And this is known as the extrinsic pathway. So the death receptor pathway is also known as the extrinsic pathway. How are these apoptotic cells cleared? Now the cells undergo several changes in the membranes, and this helps to promote phagocytosis. Now the normal cell contains phosphatidylserine in the inner leaflet of the plasma membrane. In the apoptotic cells, this phosphatidylserine is then flipped to the outer membrane, and this is then recognized by the macrophages, and it then undergoes engulfment by the phagocytes. Another theory which has been suggested is that the cells dying by apoptosis secrete certain soluble factors which recruit macrophages. Okay, So in the first figure, you see that there is an externalization of phosphatidyl serine. That is, it's flipping from the inner leaflet to the plasma membrane. The phosphatidyl serine flips to the from the inner leaflet to the plasma membrane, and this is then recognized by macrophages, and there is engulfment by the phagocytes. What are the examples of cells undergoing apoptosis? Firstly, growth factor deprivation can result in cells undergoing apoptosis. For example, lymphocytes which are not stimulated by antigens or cytokines, neurons which have been deprived of the nerve growth factor. Second, DNA damage, that is, exposure of cells to radiation, chemotherapeutic agents, induces severe DNA damage which is irreparable and can trigger apoptosis. Accumulation of misfolded proteins. These misfolded proteins are then targets for proteolysis. Then you have the apoptosis of self-reactive lymphocytes. So if these lymphocytes encounter self-antigens, they undergo apoptosis. And lastly, cytotoxic T lymphocyte mediated apoptosis. So in this particular figure, you see a DNA which has been damaged, and this cannot be repaired. So this DNA which has been damaged and the DNA which is irreparable then is directed towards apoptosis by activation of p53. Now if on the other hand p53 is suppressed, then what happens, and the BCL6 is activated, then the cell is able to survive and proliferate, and this can lead to cancer. Thank you.